Hey everyone! So today we're tackling something a lot of people have experienced, but maybe can't quite explain, the chiropractic adjustment. We're going to go way beyond just the feeling of relief. We're diving deep into the hard science to see what's actually happening inside your brain and nervous system.
It all comes down to this one big question, right? When a chiropractor adjusts your neck and you hear that pop, what's going on at a neurological level? Is it just a sensation, or is something really measurably changing in your brain's function? Well, that's the mystery we are unpacking today.
For a long time, the science behind this was kind of a black box. Yeah, we've studied patient results, but the how, the neurophysiological mechanics of it all, that's been a lot harder to pin down. But a really interesting study from 2007 decided it was time to finally take a look inside.
So, to solve this puzzle, the researchers needed a way to listen in on that conversation happening between the body and the brain. How do you actually peek inside the nervous system in real time to see what an adjustment does? The answer, it turns out, is a technique called SEPs.
Okay, Somatosensory Evoked Potentials. That sounds super complicated, I know, but the idea is actually pretty simple. Think of it like this. You send a little ping from a nerve in your arm, and then you use some electrodes on the scalp to listen for the brain's echo. It lets scientists measure, with incredible precision, how the brain is handling that sensory signal.
So here's the play-by-play. First, they stimulate the median nerve at the wrist with a tiny, totally harmless electrical pulse. That signal then zips all the way up the arm, through the spinal cord, and right into the sensory part of the brain. And the whole time, electrodes on the scalp are recording the brain's response, which shows up as a very distinct brainwave.
Now, here's where good science comes in. To get results you can trust, you can't just test the adjustment by itself. You absolutely need a control group, something to compare it to. This makes sure that any changes you see are actually from the manipulation and not something else. The study design here is key.
So the researchers set up two groups. The first one got a standard cervical spine manipulation. You know, that quick, specific thrust? But the control group, they had their head moved into the exact same position, but they didn't get the final thrust. It's a brilliant way to isolate the specific effect of the adjustment itself.
And who were they testing? Well, all the participants had a history of neck stiffness. But, and this is a really important detail, they weren't in any pain during the experiment. This was on purpose. They wanted to take pain out of the equation, since pain itself can change brain activity. They were just looking at the effect of manipulation on dysfunction, period.
All right, so the stage is set. We've got the measurement tool, we've got the groups, and the experiment is underway. So what happened? What did the brain signals look like before and after? This is where it gets really interesting.
The study's conclusion says it all. The manipulation led to transient cortical plastic changes. Okay, that's a mouthful, so let's break it down. Transient just means it was temporary. Cortical plastic changes means the brain's cortex, the outer layer, was actually changing how it worked. And in this case, the brain's response, its echo, was getting weaker or attenuated.
And you can see it right here in the data. Look at the top chart A. That's our control group. The bars before and after, they're pretty much the same. But now look at the bottom chart B, the manipulation group. See those bars with the stars on them? In science speak, that's like a big flashing sign that says, hey, this is a real, statistically significant change. And if you're a visual person, this slide makes it crystal clear. On the right, in the control group, the before and after brainwaves are basically carbon copies. But look to the left at the manipulation group. Those peaks called N20 and N30, they are obviously smaller, flatter after the adjustment. You are literally looking at a change in brain activity.
So just how big of a change are we talking about? Well, that N30 peak, which is tied to how the brain uses sensory info to plan movement, it showed the biggest change. It dropped by an average of over 30% in just the first 10 minutes after the adjustment. That's a huge shift.
And here's the kicker. This is how they knew for sure this change was happening in the brain. They also measured the nerve signal on its way up, at the arm and at the spine. And those signals, totally unchanged. The ping was just as strong as before. It was the brain's echo, its processing of the signal, that had changed.
Okay, so a temporary 30% shift in brain processing is pretty wild. It's a fascinating scientific finding, but what does it actually mean? Let's zoom out and talk about the bigger picture and why this change in the brain is such a big deal.
So let's wrap this up with the key takeaways. First, a physical adjustment can cause real, measurable changes in how your brain processes information. And while the effect in this study was temporary, it gives us a potential neurological reason for why some patients feel and function better.
It suggests that the adjustment might be altering how the brain pieces together sensory information. This is huge.
At its core, this whole thing is about sensorimotor integration. All that means is it's the process your brain uses to take in all the sensory stuff from your body and use it to plan and control your movements. What this study suggests is that a spinal manipulation might help to sorta reset that entire feedback loop.
And that leaves us with a pretty incredible question to think about. We now have evidence showing a clear link between a physical action at the spine and a real functional change in the brain. So, if a single adjustment can temporarily change how the brain works, what could that mean for the future of all kinds of therapies and for our whole understanding of that powerful brain-body connection?